I'm David Yankelevitz, and I will be leading the session on big data and vulnerable populations addressing the gap. These are financial and research disclosures. For this session, which I'll be also sharing with Chris Latham in the accompanying hour, but for my session, these are the panelists, and they'll introduce themselves during the discussion. In order to think about this topic, we're thinking about the pathway to development of a useful database, and we're focusing here on low-dose CT in the context of lung cancer screening. I think we need to define the purpose of the images. What are we collecting data for? Is it for advanced image processing or visualization? Is it for quantitation? Is it for detection? And I think, at least to an extent, you want to focus on what you're looking to do with the database. Is it for lung cancer? Is it for emphysema? Is it coronary calcium, osteoporosis? Or do you really need to define it completely, because as time goes on, if it's a good database, you'll use it for other health measures that may be available in the images that we're not even aware of. But in each case, the necessary quality needs to be defined. And I think that's a very important aspect of having a database and making it valuable. This is an article that we published a few years ago. This was tumor volume measurements error in the context of a clinical trial. This was a lung cancer trial done with Roche, and we were measuring image quality. And this is from calibration devices that were placed on the patient to see if we could measure things like we hoped for. And A, B, and C are transaxial, sagittal, and coronal views of the Teflon sphere. It's about a little more than 1.5 centimeters. And you can see, at least on that initial scan, that was all done at the same time, looks about the same for the sphere. But then a second follow-up scan was obtained, that's D, and you can see suddenly that sphere in the coronal view suddenly looks a lot smaller. That's the purple line. And then in another subsequent scan, E, it got a lot bigger. Now the calibration sphere didn't change, but clearly there's some distortion here. And finally, slide F, or F here, shows that you can see the green represents kind of the XY plane, and that didn't change very much. But along the poles, north and south, there's a lot of variability in that calibration phantom. And you can see what this does in terms of volume measurement. It turns out in this study, there were 3 manufacturers. And for manufacturer A, you can see the proportional change in volume. And this occurred with 16 slice or less scans. It's enormous. If you're thinking about measuring something, a small nodule, this basically makes it unusable. They seem to have corrected the problem to a large extent for their greater than 16 slice scanners. But you can look at other manufacturers. Manufacturer C had much less of a problem for 16 slice or less. But for 16 slice or more, there's still a fair amount of error. And when you think about applying this to a 5 millimeter nodule, that percent error may be quite impactful on our ability to measure a small nodule. So these are things that we have to think about. What kind of quality do we need in these databases? And I just show you another example of this. This comes from an experiment that I was involved with, where we looked at 3 rolls of Scotch tape from the center of the field of view out to the periphery with a particular scanner that had that distortion. And just look at that roll of Scotch tape, what happens to it. Imagine if that was a small lung nodule. And finally, I just show this. This is a surface rendered view of a patient, and you can guess which scanner this was on, giving these kind of ripple effects or distortions. Now, beyond the idea of the image quality, we also have to think about the databases and what we're measuring and the size of the database and how useful they are. And I'm just showing, for example, this is in trying to predict whether something is lung cancer. And you can see this group gave ROC curves. They're doing methylation, and they're using particular databases, and they have an area under the curve of 0.76 up to 0.87, depending on whether they combine clinical data with the methylation. And here's another group, and they're using a different process, and they have an area under the curve of 0.73. Another group, and everybody is producing these kind of images, but they're all using different databases with different size cancers. And finally, this comes from Median Technology, which used the NLST database, and they got spectacular performance for predicting whether something is malignant, and they actually come up with an area under the curve that's at 0.99. The question is, how well does this do? Would it do on the other databases, or how well will those other procedures do on this database? And here's where we really need to get larger databases together, because there's really a problem with these small databases and everybody doing something differently, and whether you're producing anything meaningful really at all. Now, to address this problem, a group has been formed called MIDRC. This is a multi-group 2-year study. It includes the AAPM, ACR, and RSNA, as well as other institutions. And it stands for the Medical Imaging and Data Resource Center. And it's very much been built to accelerate development of AI. And some of these slides that I'm now showing were given to me by Mary Ellen Geiger from the University of Chicago, who's the PI of this project. And you can see all the organizations that are involved and some of the people, very prestigious group. And they have multiple subcommittees, and they've really thought about a lot of the problems for how to collect data, how to distribute data, both on the technical side, the ethical side, and they've done a lot of work in moving towards making this available. They've thought about things like getting good representation of diversity, and they've sort of tagged it to the population of the census data, and you can see census data in blue and MIDRC data in orange, what they're doing in terms of populations. They've also thought about some of the ethics, and this is an article that they reference, which talks about the purpose of data and whether it really should be usable. Once it's finished its diagnostic purposes, can institutions share the data? And MIDRC has done a lot of work in terms of developing data use forms and data use agreements that they're making available to institutions to make it easier for them to share their information. I wanted to give you an example of why big data is so, I think, useful. This came out of our LCAP work. And in this paper, we looked at over 50,000 patients to see the amount of COPD. And it turned out that somewhere around 25% of the people had COPD. And among those 25%, almost 3/4 of them didn't even know that they had emphysema. I mean, it's really an extraordinary number. And amongst those, there's a fairly high percentage that actually had severe emphysema and had no idea that they had it. This is some of the things you can do with these large databases. So there's a lot to think about here in terms of making databases useful and some of the challenges. And I just want to throw a few things out there. You know, we have lots of legacy databases, and how they're used can be a challenge. In particular, I think about the coronary artery calcification databases, where they were built using 3mm slice thicknesses. And they've really become entrenched. It's very hard to get cardiologists to think of anything other than those 3mm slice thicknesses because they've used it for so long. But nowadays, we're using much thinner slices. And there's more information in those thin slices. One of the most important things in coronary calcium scoring is a 0 score. But a 0 score on a 3mm slice might not be 0 when you use a thinner slice. The NLST is available, tremendous resource. But those scans are, to some extent, obsolete. 2.5mm slice thickness now is no longer the state of the art. And we have similar challenges with osteoporosis databases, where the databases were built primarily using premenopausal women and then extrapolated to other populations. We have challenges with combining information from different databases. It's not just imaging. We want to include molecular data, genetic, proteomic. How do we do this? These databases aren't all together. And there are mechanisms for doing this, extracting information from the electronic health record. How do we do this? MIDRC has addressed some of these issues, but there's much more to do here. And one of the other problems or challenges really is how do you establish normal values for diverse populations? Certainly the tools for what we measure are the same in these diverse populations. A nodule size is the same regardless of what the population is, but whether that's a normal value or whether other things that we measure, like bone density, is the same for diverse populations. How do we define these populations? How do we define normal values? And similarly, if we want to build a database that's useful, how do we determine the number of participants so that we can come up with a normal value for these various propositions that we want to measure? How do we obtain data? Well, what are the needs? And here we have to think about the needs of the academic institution as well as societal needs. And this is going to be a topic of great concern. Academic institutions just can't typically give away large amounts of data because there's a cost associated with it. And similarly, there are societal needs for the data. And finally, who gets access? If we build these kind of databases, who has access to them? I know I was personally involved in a situation where LCAP at one point was subpoenaed by Philip Morris because Philip Morris was involved in class action lawsuits to support screening. And they felt they needed to analyze the data from LCAP more carefully than what was available in our published works, and they wanted access to it. But when patients that were, or participants at LCAP found out that Philip Morris might have access to their data, they were already threatening to withdraw from the study. So who gets access to data? Other people have this feeling when it comes to pharmaceutical industry getting their data. So there's lots of considerations about who has access. Does industry have access? And there are many approaches for this. When you build these databases, sequestering certain amounts of data so that it can be used for FDA. These are all considerations that hopefully we'll address in the panel discussion. Thank you.